This week in medical entomology, the most talked about mosquitoes were the ones that cannot bite you. Male mosquitoes do not take blood, only females do, and that is exactly why a male is such a useful thing to release. In Singapore, they have been released by the million. The program is called Project Wolbachia. Wolbachia is a bacterium. When a male carrying it mates with a wild female who does not, her eggs are fertilized and then they simply stop. They never hatch. This week, the New England Journal of Medicine published the suppression results. Dengue came down where those males went out. The same issue carried a reply from the Singapore team. That is what a serious result looks like. It gets argued with in public by name. Now, the harder question, where does dengue virus wait between outbreaks? One answer arrived as a preprint. They found dengue virus inside Aedes aegypti eggs in the field, not in a cage, in natural settings. That is vertical transmission, mother to offspring, before the larva has ever met a human. And an Aedes egg can survive dry for months. If the virus rides along inside it, the dry season is not a break in transmission. It is a pause button, which brings us to the chemicals. A modelling study asks what resistance evolution actually does to a malaria control program over time. The uncomfortable part is that the insecticide keeps working right up until it doesn't, so the field is getting better at watching for it. A new droplet digital PCR assay screens Aedes albopictus for KDR mutations. Those are changes in the sodium channel that pyrethroids are supposed to hit. Others went at it from the dose side. Resistance intensity testing in Aedes aegypti asks not just whether she survives, but how much it takes to kill her. A separate team designed the first primers for that same sodium channel in the kissing bug that carries Chagas disease. If the insecticides are wearing out, what replaces them? In Burkina Faso, one answer is being tested in the blood itself, a long-lasting ivermectin formulation in a semi-field trial. The idea is simple and slightly brutal. The drug sits in the host, the mosquito bites, and the blood meal kills her, which is why cattle keep turning up in malaria papers. A spatial analysis looked at how the presence of cattle changes malaria risk for the people living beside them. And here is a warning for anyone who reads trial results. One study changed only the architecture of the experimental huts, and the entomological endpoints moved. Same nets, different building, different answer. Now geography. In Kenya, researchers made the case for prioritizing urban water storage containers in the hunt for Anopheles stephensi. Stephensi is the invader that broke the rules. Classic African malaria vectors breed in puddles and rice fields. This one breeds in the tank behind your house, in the middle of a city. In China, the picture is almost the opposite. Yunnan province reported 5 years of imported malaria along the border with Myanmar. Elimination inside a border does not stop a parasite arriving across one in Bangladesh. A nationwide analysis found urban hyperendemic dengue and a rising per head risk along the coast. In Brazil, 10 years of surveillance mapped where Aedes aegypti and Aedes albopictus have each settled across the state of Santa Catarina. Aedes aegypti is the one to know. Small, dark, with pale banded legs. It lives with people, it breeds in what we leave outside, and it bites in daylight. In Iran, a review told a two-part story. Aedes aegypti disappeared, then it came back, and with it a rising dengue threat. And in Indonesia, the CDC issued a level 2 travel notice for Zika this week on the 7th of August. Level 2 means practice enhanced precautions. For Zika, the people that notice is really written for are pregnant travellers and anyone planning a pregnancy. Elsewhere, a dose-response study found that West Nile virus lineages 1 and 2 do not behave alike. Same virus name, different behaviour at the same dose, that matters right now. In Albania, seroprevalence in horses is rising. A horse is a sentinel because horses and people are both dead ends bitten by the same mosquitoes, and those mosquitoes are Culex, not Aedes, a night-biting, bird-feeding genus most people have never heard of and have definitely been bitten by. On the tick side, a team built an image-based tool for tick species identification point, a camera. Getter species identification has always been the bottleneck. And in Japan, genomics compared native populations of the Asian longhorn tick with the introduced ones now living in Oceania. This is a tick that can reproduce without a male, one female, one host, one founding population. That is how a species crosses an ocean and stays in the eastern Algerian Sahara. Serology in dromedary camels found 3 arthropod-borne viruses circulating quietly: Rift Valley fever, West Nile fever, and Crimean Congo hemorrhagic fever. Hidden circulation is the phrase the authors used. No outbreak, no cases, just antibodies in animals nobody was testing. Finally forward, one paper set out what post-release environmental safety monitoring should look like for gene-drive mosquitoes. These are the questions you must be able to answer before you let a gene spread on its own. And a reminder that not every threat is a mosquito. Screwworm work continued this week with a standardized test of 4 compounds against the larvae, a fly whose maggots feed on living flesh and which is once again on the move. 86 papers crossed the desk this week. That was the shape of them. Links to everything are below. If this is your kind of thing, subscribe and I will see you next week.